You have learned about electron carriers that allow the flow of electrons through the electron transport chain. And I made an allusion of 4 protein complexes that are containing these electron carriers. So in this video, we will take a closer look at the transport of electrons within these 4 complexes. And you will learn that each complex uses a different mechanism to transfer electrons. Let's start with complex 1. Complex 1 is the first entry point to the electron transport chain. Complex 1 has an L-shaped structure with an arm which is found in the mitochondrial matrix and an elongated structure that is embedded in the inner membrane of the mitochondria. It's a very complex multi-subunit molecule that contains about 42 subunits. Its function is to pass electrons from NADH to coenzyme Q. Hence its name, NADH ubiquinone oxidoreductase. But we'll call it complex 1. NADH will interact with a subunit of complex 1 that is found in the arm part of the complex and contains an FMN universal electron acceptor. The electron from NADH will be transferred one at a time to form first FMNH and then FMNH2. The electrons and protons that are captured by FMN are passed along multiple carriers located in close proximity such that the electron transfer is possible. First, FMNH2 will give its electrons to a protein that contains an iron-sulfur center. Then this electron will move to an iron-sulfur protein and finally to coenzyme Q. And then the 2 protons that were bound to FMNH2 are directly transferred to coenzyme Q to form ubiquinol QH2. The transfer of electrons through complex 1 is also coupled with the extraction of 4 additional protons from the mitochondrial matrix that are moved into the intermembrane space. Now complex 2. Complex 2 is the second point of entry to the electron transport chain. While NADH is the source of electrons for complex 1, FADH2 is the source of electrons for complex 2. Complex 2 is also called succinate dehydrogenase. We have already encountered this enzyme, which catalyzes a step of the citric acid cycle. So this enzyme contributes both to oxidative phosphorylation and to the citric acid cycle. In the citric acid cycle, the succinate dehydrogenase catalyzes the conversion of succinate into fumarate. One of the subunits of the enzyme contains FAD. So during the oxidation of succinate, electrons and protons will be transferred to FAD to form FADH2. Complex 2 is very important because it allows the direct coupling of citric acid cycle with oxidative phosphorylation. So let's try to figure out how complex 2 transfers its electrons from FADH2 to coenzyme Q. Complex 2 functions as a trimer with a mushroom-like shape, but we will focus on one of the monomers and learn how electrons move through this monomer. On the structure that is presented here, you can see that complex 2 is formed of 4 subunits. 2 reside in the mitochondrial matrix and 2 are embedded in the inner membrane. FADH2, which is bound to the SDH-A subunit, will transfer its electrons to a series of iron-sulfur center contained in the SDH-B subunit, and then these electrons will be transferred either directly to coenzyme Q, which is attached to SDH-D, or to a heme, which is contained in SDH-C, and then to coenzyme Q. And then the two protons of FADH2 that are released during the oxidation of FADH2 will react with coenzyme Q to form ubiquinol. If we take a closer look at the electron carriers that are found in a monomer, we can see that they are stacked on top of each other. And the edge-to-edge distance between two carriers is always less than 40 angstrom, which is a suggested limit for the physiological transfer of electrons. Therefore, the electrons can easily hop from one carrier to another carrier. The standard reduction potential of the different carriers that are contained into complex 2 determine the unidirectional path of electrons from FADH2 to coenzyme Q. In fact, the proximity of coenzyme Q to the last iron-sulfur center and the difference in standard reduction potential of coenzyme Q and the heme B favors a direct transfer of electrons from the last iron center to coenzyme Q. So in summary, complex 1 receives electrons and protons from NADH and passes them to coenzyme Q. Complex 2 receives electrons and protons from FADH2 and passes them to coenzyme Q. As you can see, coenzyme Q plays a central role in collecting electrons and protons from universal donors. But in fact, coenzyme Q accepts also electrons and protons from other sources. On the mitochondrial side, the first step of the oxidation of fatty acid is catalyzed by an acetyl coenzyme A dehydrogenase. This enzyme contains a molecule of FAD that will get reduced in FADH2, and then FADH2 will pass its electrons and protons to coenzyme Q via a couple of intermediate transporters. Glycerol 3-phosphate, which is formed in the cytoplasm from glycerol or from dihydroxyacetone phosphate, one of the intermediates of glycolysis, will diffuse inside the intermembrane space and will then be converted back into dihydroxyacetone phosphate by the mitochondrial glycerol 3-phosphate dehydrogenase. This reaction is coupled with the reduction of FAD that is bound to the hydrogenase. FADH2 will then pass its electron and protons to coenzyme Q. Now, what is the structure and the function of complex 3? So first, the function of complex 3 is the transfer of electrons from ubiquinol, QH2, to cytochrome C, which is attached to the surface of the inner membrane of the mitochondria on the intermembrane side. Cytochrome C, in fact, can bind to the part of complex 3 that is emerging from the inner membrane on the intermembrane side. Complex 3 acts as a dimer, and the monomer, which is shown on this structure, contains 11 different subunits. But we'll focus on 3 subunits that are represented in color on this structure that form the functional core of complex 3. The first subunit is cytochrome C1 that will interact with cytochrome C. The second subunit is a protein that contains a risky type iron-sulfur center. And the third subunit is cytochrome B, which is embedded in the inner membrane of the mitochondria, contains 2 hemes, as well as 2 binding sites for coenzyme Q, called Q0 and Qi. The electron transfer from coenzyme Q to cytochrome poses a challenge. Indeed, coenzyme Q can donate 2 electrons, but cytochrome C can only accept 1 electron. Complex 3 solves this problem by having 2 separate paths for the electrons that will lead to the production of 2 molecules of reduced cytochrome C. The Q cycle provides also a mechanism to transport additional protons across the inner membrane of the mitochondria. So how does the Q cycle work? It's a multi-step process. In the first step, complex 3 has a molecule of oxidized cytochrome C that is bound to cytochrome C1. Its Q0 site is occupied by ubiquinol, the fully reduced coenzyme Q, QH2, and its Qi site is occupied by a fully oxidized molecule of coenzyme Q. 1 electron from QH2 on Q0 will be transferred to cytochrome C via an iron-sulfur center and then the heme from cytochrome C1. The second electron will be transferred to the heme of cytochrome Bl, then to the heme of cytochrome Bh, and finally to the oxidized coenzyme Q bound on Qi to form a semiquinone. The 2 protons from ubiquinol on Q0 will be released in the intermembrane space. So at that point, what we have is a complex 3 that has just reduced a molecule of cytochrome C, and the cytochrome C has been released. The Q0 site is now occupied by a fully oxidized molecule of coenzyme Q, and the Qi site is occupied by a semiquinone. The fully oxidized coenzyme Q dissociates from Q0, is replaced by a fully reduced molecule of coenzyme Q, QH2. And then a new molecule of oxidized cytochrome C binds to cytochrome C1. So now that a new molecule of QH2 is bound to Q0, as it was described before, 1 electron will move upward to reduce the molecule of cytochrome C, and this reduced cytochrome C will diffuse away. And 1 electron will move downward to the semiquinone Q that occupies the Qi site. And this electron plus 2 protons that are extracted from the mitochondrial matrix will form a fully reduced molecule of coenzyme Q, QH2, that is bound to the Qi site. Then the 2 protons from the fully reduced coenzyme Q that was bound to Q0 will be released in the cytoplasm. So in the third step, the fully oxidized coenzyme Q, which is bound to Q0 will dissociate, and the fully reduced coenzyme Q, QH2, which is bound to the QI site, will dissociate as well. And the last step is just to reset the Q cycle with one molecule of fully reduced coenzyme Q that binds to the Q0 site, one molecule of fully oxidized coenzyme Q bind the QI site, and a new molecule of cytochrome C that is oxidized can bind to complex 3. So overall, only one molecule of fully reduced coenzyme Q, QH2, gets consumed, 2 cytochrome C gets reduced, and 4 protons are transferred across the membrane. What is now the structure and the function of complex 4? Well, complex 4 will transfer electrons from reduced cytochrome C to the final electron acceptor, oxygen, to form water. The structure that is presented here of complex 4 shows that complex 4 acts as a dimer. And each monomer will contain 13 subunits. But we will focus on 3 subunits that are represented on this structure in color that represent the functional core of complex 4. The transfer of electrons from reduced cytochrome C to oxygen is a multi-step process. Step 1. One molecule of reduced cytochrome C binds to complex 4, and then will pass its electron through a series of carriers. A copper center, QA, then a heme, A, then another heme, A3, and finally the last copper center, QB. So the cupric ion that is bound to QB, Cu2+, will get reduced. Step 2. A second molecule of reduced cytochrome C bind to complex 4 and replace the previously oxidized cytochrome C, will pass its electron through the same path. But when this electron reaches heme A3, it cannot be transferred to the QB center because the QB center is already reduced. So now the heme A3 will be reduced, and more specifically, the ferric ion, Fe3+, carried by the heme A3, will be reduced into a ferrous ion, Fe2+. But do you remember what happened in hemoglobin? In the heme of hemoglobin that has an Fe2+ ion, this heme can bind oxygen. That is exactly what happened with complex 4. Once the heme A3 is reduced, then an oxygen molecule can bind to the ion of A3. But we have the reduced copper ion that is very close. And in fact, this ion can transfer its electron to oxygen. And the consequence is the formation of a peroxide that covalently linked the ion and the copper via the oxygen molecule. Step 3. A third reduced cytochrome C binds to complex 4, passes its electron through the same path, and this electron plus a proton that is extracted from the mitochondrial matrix will lead to the cleavage of the peroxide bond. One oxygen atom will be bound to iron, and the hydroxyl group that is formed will be bound to copper. Step 4. A fourth cytochrome C in its reduced form bind to complex 4, passes its electrons through the same path. And this electron, in combination with another proton that is extracted from the mitochondrial matrix, will lead to the formation of an hydroxyl group that is attached to iron. Now we have an hydroxyl group bound to iron and an hydroxyl group bound to copper. Then two other protons extracted from the mitochondrial matrix will come in, react with the two hydroxyl group to form two molecules of water. And now complex 4 is ready for a new transfer and a new round that will lead to the formation of water. So overall, 4 reduced cytochrome C are consumed, are oxidized. 1 molecule of oxygen is converted into 2 molecules of water. And 8 protons from the matrix are consumed. 4 contribute to the formation of 2 molecules of water, and during the transfer of electron, 4 additional protons are transported across the membrane from the mitochondrial matrix to the inner membrane state. So in conclusion, the 2 electrons and 2 protons given by NADH plus H plus lead to the formation of 1 molecule of water and the transport across the inner membrane of the mitochondria of 10 protons that will lead to the buildup of the proton gradient.
I thought that complex 4 produced 2 water molecules, but you just said that 1 water was produced. Did you make a mistake?
No, I didn't make a mistake. In fact, both of my statements are correct. You have to carefully count the different molecules. So if we go back, 1 NADH plus H plus produces 2 electrons and 2 protons. These 2 electrons are responsible during the Q cycle for the formation of 2 reduced cytochrome C. However, to form 2 molecules of water in complex 4, you need not 2, but you need 4 molecules of reduced cytochrome C. And these will come from the oxidation not of 1, but 2 molecules of NADH plus H plus. Therefore, the 2 electrons and protons from 1 molecule of NADH consume half of a molecule of oxygen and produce 1 molecule of water.